Hello everyone. In this video lecture, we are going to discuss about the plasma membrane. Okay, from the topic cell, the unit of life. So let's discuss the plasma membrane in a bit detail. Now, when we talk about plasma membrane, a cell, okay, which is a basic unit of life, it has a border that is made up of this membrane, which is called as a plasma membrane. It may also be called as a cell membrane. It is also called as plasma lemma. Okay, so this is the different names that we can call for a plasma membrane. Now, when we talk about plasma membrane, the detailed structure of this membrane was studied after the advanced discovery of an electron microscope in the year 1950. Okay, somewhere in 1950s, and the chemical studies have been done on the cell membrane, especially on the human RBCs. So based on the facts that have been studied on RBC, and with the help of electron microscopy in 1950, we get this structure of the plasma membrane. Okay, the studies have shown that the membrane is made up of two layers of the lipids. Okay, how many layers of the lipids? Two layers of the lipids are present, and it is as the two layers are present, it is also called as the bilayer. Okay, so two lipid layers are present. So let us draw here some uh, representative diagram over here. So we can see these head-like structures. Okay, these head are basically the phosphate head, and the tails which we can see, those are the lipid tails. Okay, so one single structure is made up of a phosphate head. Okay, and the tail is called as a lipid tail. Phosphate is having a negative charge on it, so the head is polar in nature, whereas the tail is non-polar in nature, does not have any charge on it. Coming to the next point is that the phosphate head is hydrophilic, that is, it loves water, whereas the lipid tail, it is hydrophobic in nature, that it does not like the water. Okay, so in this way, the two layers of these phospholipids are making up the maximum part of the plasma membrane. Okay, and these two layer will be called as a phospholipid bilayer. Phospholipid bilayer. Okay, so these are the basic things, but when we talk about this lipid and the phospholipid bilayer and everything, in between these phospholipid bilayer, you can see in the diagram that there are these blue color structures which are present. These are basically the proteins that are present. It is found that around 50 to 52% is protein in the plasma membrane, and 40% of the plasma membrane is made up of the lipids. Okay, so you can see here there are two types of proteins. If we have to consider the proteins, we can categorize the proteins into two different categories. One category is called as the intrinsic type of proteins. Another proteins are called as the extrinsic type of proteins. What are these intrinsic proteins? The proteins which are completely inserted into the plasma lipid layer. Okay, now you can see over here, this is a protein which is present on the plasma membrane, and it is completely interdigitated in the plasma membrane, passing through the plasma membrane, and is present on both the sides. So this is called as an intrinsic or integral protein. But when we talk about some extrinsic proteins, they are just superficially attached on the surface, whether it might be the upper surface or whether it might be the lower surface. So these are called as a surface protein, or they are called as the extrinsic protein, or they are also called as the peripheral proteins. When we talk about the intrinsic proteins, these proteins are very difficult to isolate or to separate them from the plasma membrane with the help of the detergents and so on, with those kind of treatments. If you try to remove the intrinsic proteins, if you try to separate the intrinsic proteins, the plasma membrane, they get completely may get damaged. That is not in case, that is not the case in case of the extrinsic proteins. Extrinsic proteins can be easily separated, okay, from the plasma membrane without disturbing or without distorting the plasma membrane. So these are about the two types of proteins that are present. Now, when we talk about some of the scientists, they have given the models of the plasma membrane. One model was given by Danieli. One model was given by Danieli and Davison. Danieli and Davison, but it is not widely accepted. Okay, they said that the plasma membrane is made up of four layers. Okay, two layers are of lipids and two layers are of proteins, but not widely accepted. Later on, Robertson gave one model for the plasma membrane. He stated that there are two layers of the lipid which are covered by proteins. Okay, so that was given by Robertson. But the widely accepted model for the plasma membrane is a model which is called as the fluid mosaic model. Fluid mosaic model, which was given by two scientists, Singer and Nicholson. So this is the most widely accepted model of the plasma membrane, and it was given in the year 1972. Now why it is called as fluid mosaic model? Fluid, that is, they consider the plasma membrane as the fluid. It gives fluidity to the plasma membrane. And what is mosaic? Mosaic are the patches. Okay, so if there is a sea of lipids, in this sea of lipids, you can see some patches of proteins. So these are the mosaics of proteins. So that's why the model is named as the fluid mosaic model, which was given by Singer and Nicholson. Okay, so here when we talk about the quasi-fluid nature of the lipids, okay, because of the quasi-fluid nature of the lipids, the proteins can move from one place to another in this membrane. Okay, the protein can have the lateral movements they can have. That is, suppose if this is a protein, yeah, it can move in this direction or it can move in this direction, right? So this is what is called as the fluidity of the membrane. Okay, this ability where the proteins can laterally move in the quasi-fluid lipid layer is called as the fluidity of the membrane. Okay, now the fluid nature of the membrane is very important. Because of the fluid nature of the membrane, the functions like cell growth, okay, can take place. The functions like formation of the intercellular junctions, that is, the junctions between the two cells, can happen because the plasma membrane is found to be fluid. Okay, it has the fluidity. Then the secretion, suppose if some cells, okay, synthesize some substances, they have to be secreted outside the cell. So for that purpose, vesicle formation is very important, and these vesicles, they bud out from the plasma membrane and they take out all these substances outside the cell. And then there is a function which is called as endocytosis. Okay, endocytosis is something where a cell, when it comes in contact with another substance or any other cell, it can engulf that cell inside the cytoplasm. This is called as endocytosis, and once after the engulfment, it may destroy that inside the cytoplasm. And also it is important for the cell division. Okay, so the fluid nature of the plasma membrane has been seen to be positive in this way, because it is going to help for all of these functions. Right, let us talk about the integral proteins. On the plasma membrane, 70% of the proteins which are found, these 70% of the proteins are found to be the integral proteins. Okay, and they can't be extracted, we have already told. These integral proteins, they may form the tunnel proteins. What are the tunnel proteins? If suppose if this is a plasma membrane, okay, they form a tunnel like this, through which the solutes, okay, the charged solutes which cannot travel directly from the lipid membrane, okay, they can be transported through this tunnel proteins, or the channel proteins we can also call. These integral proteins, they also form transmembrane proteins. Transmembrane, that is these are called as the transmembrane proteins. Okay, very much important during the time of cell signaling, right? So these are the transmembrane proteins. One example of such integral protein is glycophorin. Okay, so glycophorin. Whereas when we talk about the extrinsic proteins, extrinsic protein example can be spectrin. Okay, it is an extrinsic protein which is just superficially attached to the plasma membrane on the surface. Okay, now these extrinsic proteins, they can be attached to the plasma membrane covalently, okay, or they might be attached non-covalently, okay, to the plasma membrane, right? These proteins may also act as enzymes. Okay, these proteins which are present on the plasma membrane, they may also act as enzymes, and one such enzyme is permease. As the name suggests, permease, it is going to allow the permeability, it is going to increase the permeability of the plasma membrane. What is permeability? The plasma membrane is selectively permeable. Okay, it only selects some of the substances they can pass across the plasma membrane. So this permease can increase the permeability of the plasma membrane and can allow the entry of some substances inside the cell or exit of some substances outside the cell. Okay, now the plasma membrane is all about this. Let us talk about the functions of the plasma membrane. The plasma membrane, as I said, is selectively permeable. It is going to select for those molecules to enter and to pass across it. Okay, when we when we talk about the transport across the plasma membrane, one transport is called as diffusion. Okay, we can call it as passive diffusion. Okay, or we can call it as a passive transport. Right, so what is diffusion? Basically, diffusion is the transport of the solutes which are basically non-polar. They do not carry any charge. Okay, so they can cross the plasma membrane from their higher concentration gradient towards their lower concentration gradient. Okay, the gases, they also carry out diffusion process. Solutes which are non-polar, they also carry out the diffusion process, which is a passive transport. There is no requirement of any carrier protein over here, right? And there is no requirement of any energy in the form of ATP over here. So this is called as a diffusion. When we talk about the transport of water, water also diffuses, but the diffusion of water is called as osmosis. Water molecules will move from their higher concentration gradient towards their lower concentration gradient always. Okay, so this is called as a diffusion, osmosis. Let us talk about one more type of a transport, which is called as the which is called as a facilitated diffusion. What is this facilitated diffusion? Okay, we know the structure of plasma membrane and there are some integral proteins, right? So if there is any ion or if there is any substance that has a charge on it, it cannot directly pass from the lipid layer. It has to be transported with the help of a carrier protein. Okay, this carrier protein helps this charged solute to get transported from one side of the plasma membrane to another side of the plasma membrane. Okay, it can be vice versa, right? So only carrier protein is required, no ATP is required over here, and the transport is from higher concentration gradient to lower concentration gradient. Okay, this is called as a facilitated diffusion. And the last type of a transport is called as active transport. What is active transport? Again, there is a plasma membrane, right? And there are some channels, there are some protein channels. Okay, so in case of active transport, the charged ions have to be transported, but they have to be transported from their lower concentration gradient towards their higher concentration gradient. Already sodium is less here and sodium is more over here. Okay, let us consider this. So as we stated earlier, in diffusion and facilitated diffusion, always the solutes or the molecules, they move from their higher concentration gradient towards the lower concentration gradient. But here the movement is opposite. The sodium are being moved from the lower concentration gradient towards their higher concentration gradient. So for this movement, the energy is required, and it is in the form of ATP. What is ATP? It is adenosine triphosphate. Okay, the phosphatase enzyme acts on it. Okay, and this phosphatase enzyme, it breaks the phosphate bonds, because what is an ATP? It is an adenosine molecule to which three phosphates are attached, and one phosphate is broken, energy is released, it will be now called as adenosine diphosphate. If two phosphates are broken, more energy is released, and it will be now be called as AMP, that is adenosine monophosphate, right? Again, when the phosphate molecules are added, this AMP can get converted into ADP, and ADP can be get converted into ATP, and again the ATP is generated, and again this ATP can be used further for some energy consuming chemical reactions over here. The example that is given for active transport is sodium potassium pump. Okay, we will study the sodium potassium pump when we study the topic of chemical control and coordination. It is about the brain and the neurons. Okay, so on the membrane of the neurons, this sodium potassium pump is present, and it helps in transporting the signals or the impulses from one region of that axon to the another region of the axon. Okay, so this is all about the plasma membrane. In the next video lecture, we will discuss about the cell wall and the endomembrane system of the cell. So thank you so much everyone for watching it. I hope you have understood the topic. Thank you so much.